In today's video, we are going to study on calibration curve. So you start by looking at what a calibration curve is. Calibration curve is an analytical method for determining the concentration of a substance in an unknown sample by comparing the unknown to a set of standard samples of known concentrations. Let's look at some key terms that we'll be using in the preparation of the calibration curve. So the first is sample. So when we take up sample, a sample is a small portion of a larger substance or material that is taken for analysis, and the sample also contains the analyte of interest. So if you are to look at this picture over here, the orange over here happens to be our sample, which happens to be a small portion of the orange tree containing many fruits. The next is analyte. Analyte is a substance or constituent of the sample that is either quantified, identified, or analyzed in the test experiment. An example of a sample and an analyte is a sample of an orange and the amount of vitamin C being analyzed in it. We know that orange is rich in vitamin C. So the amount of vitamin C analyzed in an orange, we can say it's our analyte, and the orange happens to be our sample. Let's look at sample matrix. So when we talk of sample matrix, it just refers to all the other components of a sample other than the analyte of interest. So let's assume this is our sample over here. So these are all parts of our sample over here. These are all parts of the sample. And let's assume that this is our analyte of interest, or the vitamin C content that we are interested in. So what it means is that any other part of our orange sample apart from the analyte is known as the sample matrix. The next is serial dilution. So when we talk of serial dilution, it's just a stepwise series of dilution that are performed to reduce the concentration of a substance in a solution to a more usable concentration. And also, serial dilutions are made by performing the same dilution multiple times using certain amount of previous solutions added to the subsequent dilution. So as you can see from here, or from this picture, we prepared a lower concentration of 0.0001 molar from a full strength wastewater of concentration 1.0 molar. So to prepare this, we can first dilute our stock, which is the full strength wastewater of concentration 1.0 molar, to a lower concentration of 0.1 molar. And after that, we can also dilute the 0.1 molar to 0.01 molar, and we can also dilute the 0.01 molar to 0.001 molar before diluting it to the final concentration over here. So to do this, if you are to dilute our full strength wastewater of concentration 1 molar to 0.1 molar, we will just pick 1 ml of our stock solution and add it to 9 ml of the ionized water or distilled water to get this concentration. We will also pick 1 ml of the 0.1 molar solution and add it to 9 ml of the ionized or distilled water to prepare a concentration of 0.01 molar. And also to prepare this concentration, we pick 1 ml of the 0.01 molar concentration and add it to 9 ml of the ionized water to prepare the 0.001 molar concentration. And to prepare this concentration also, we pick 1 ml of the 0.001 molar solution and add it to 9 ml of the ionized water or distilled water to prepare the final concentration of 0.0001 molar. And these are the dilution factors for each of the dilution. So we can see that the dilution factor for this concentration is 1 divided by 10. And the dilution factor for this concentration is 1 divided by 100. This is 1 divided by 1,000, and this is 1 divided by 10,000. Let's look at the serial dilution formula. So the serial dilution formula is normally given as C1 V1 equals C2 V2, where C1 happens to be the concentration of the stock solution, and the V1 is the volume of the stock solution needed to prepare the final concentration of the solution as C2 in a final volume as V2. Let's look at standard calibration solutions. So when we talk of standard calibration solutions, they are the dilute solutions of known concentrations used in analysis to construct a calibration curve prepared from the standard or stock solution. So as we can see over here, so we can say that these various concentrations, that is 0.1, 0.01, 0.001, and 0.0001 molar over here, are dilute concentrations prepared from the full strength wastewater over here. So we can say that these dilute solutions of known concentrations are our standard calibration solutions, and we can use these diluted concentrations over here to prepare a calibration curve. So the next is standard. So when we talk of standards are solutions or materials with known concentrations of the analyte used to calibrate the instrument and construct the calibration curve. So the main importance of standards are for the calibration of the instrument and the construction of calibration curve. So one key thing that we have to note is that standards are the foundation of accurate quantitative analysis, and therefore choosing the right standard is essential for reliable results. Now let's look at choosing the appropriate standard materials. In calibration curve, we always need a standard. So first of all, what we have to notice is that the selected standard should meet the following requirement. 1, must have the same matrix as the analyte. 2, must have a very small systematic errors, example in volatilization or in volume errors, and etc. And 3, must have a high reproducibility. Let's look at blank sample. So when we talk of a blank sample is a sample containing any other things apart from the analyte, and this is normally used as blank to zero the instrument, example in spectrophotometry. Now, so if you should look at the diagram or the picture over here, we have our blank and our sample. The sample contains our absorbent, which happens to be our analyte, and the blank contains no absorbent, which is the analyte. So we are saying that a sample containing any other things apart from the analyte, so it contains all other things apart from the analyte, is termed as the blank sample. So let's start by looking at the construction of a calibration curve. These are some materials needed. We have personal protective equipment, example, the glass lab coat. You must make sure that you put on the right PPE. The next is our standard solution, the solvent, pipette, volumetric flask, UVB spectrophotometer, cuvette, and computer. So the first thing to do is to prepare a concentrated stock solution. So to do this, we prepare a concentrated stock solution of the standard by weighing the required solute and dissolving it with the required amount of solvent in a volumetric flask. The next step is to prepare standard for the calibration curve. And to do this, we perform a serial dilution. So to perform a serial dilution, we first have to label a series of volumetric flasks. A minimum of 5 standards are recommended for a good calibration curve. So that means that we need at least 5 different, 5 or more different concentrations to produce a good calibration curve. So let's just look at this example. Prepare the following concentrations: 1 molar, 0.5 molar, 0.25, and 0.125 molar in volumes of 10 from a 2 molar glucose standard solution. So first of all, what we have to note is that our standard solution is the 2 molar glucose that we are going to use to prepare these various concentrations given over here. So the equation that we are going to use is C1V1 equals C2V2 for our serial dilution. So let's see how to prepare the 1 molar dilution. So using the equation C1V1 is equal to C2V2, we make V1 the subject. So if you have to make V1 the subject from this equation, we have V1 is equal to C2V2 all divided by C1. So if you have to substitute the values into the equation, you have our V1 to be 5 mils over here. So what it means is that if you want to prepare the 1 molar glucose solution from the 2 molar glucose standard, we will pipette 5 mils of the 2 molar glucose solution and add 5 mils of distilled water to it to prepare this concentration. So let's see how to prepare the 0.5 molar concentration. Also, using the equation C1V1 equals C2V2 and making the V1 the subject, we have V1 to be equal to what? C2V2 all divided by C1. So substituting the values into the equation, we have our V1 to be 5 mils. So what it means is that to prepare the 0.5 molar glucose solution, we need 5 mils of the 1 molar glucose solution, and we add 5 mils of distilled water to it to prepare this concentration. And to prepare this concentration with the 0.25 molar, we use the equation C1V1 equals C2V2, and making V1 the subject, we have C2V2 divided by C1, and putting in the values, we have 0.25 times 10 all divided by 5, so we also have 5 mils. So what it means is that if you want to prepare the 0.25 molar glucose solution, we will pipette 5 mils of the 0.5 molar glucose solution and add 5 mils of distilled water to it to prepare this. And to prepare the concentration of 0.125 molar, we equally have to use the equation C1V1 equals C2V2, and we make V1 the subject. So if you have to make V1 the subject, we will get C2V2 all divided by C1. So putting in the values into the equation, we have our V1 to be 5 mils. So what it means is that to prepare the 0.125 molar glucose solution, we pipette 5 mils of the 0.25 molar glucose solution and add 5 mils of distilled water to it. So this is just a diagram explaining how to prepare the various concentrations as explained earlier. So you can see that this is our 2 molar glucose solution over here. So to prepare our prepare our 1 molar glucose solution in a final volume of 10 mils, we pick 5 mils of the stock, which is the 2 molar glucose, into a test tube, and we add 5 mils of distilled water to it to prepare this. Also, to prepare the 0.5 molar glucose solution, we pick 5 mils of the 1 molar glucose solution and add 5 mils of distilled water to it and mix very well to prepare this. And also to prepare the 0.25 molar concentration, we pick 5 mils of the 0.5 molar glucose solution and add 5 mils of distilled water to it and mix to prepare this. And also to prepare the final bond, which is 0.125 molar, we pick 5 mils of the 0.25 molar glucose solution prepared and add 5 mils of distilled water to it. So the next thing to do is to transfer the prepared standard samples and unknown into cuvettes. So as you can see, so these are our standards, the prepared standards that we just prepared over here in transferred into cuvettes, and these are our unknown samples also transferred into a cuvette. So the next thing to do, which is the step 3, is to measure the absorbance of standards and samples. So we measure the absorbance of each standard solution at the required wavelength with the UVB spectrophotometer. And we do same for the unknown sample containing the analyte of interest. What we also have to note is that we have to duplicate readings for each standard and unknown and record the data. So before we use the spectrophotometer or the UVB spectrophotometer, what we must ensure that the UVB spectrophotometer is well calibrated before measuring the absorbance. And we normally use the standard to calibrate the UVB spectrophotometer. So the fourth step is to plot the calibration curve. So to plot a calibration curve, we plot the data with absorbance of the standard solutions on the y-axis and the unknown concentrations on the x-axis by the help of a computer. And normally, the calibration curve looks linear in this form like this. And this is the equation of the line, which is always given as y equals ms plus b, and this is our r square. So the next thing to do is to fit the data to a linear regression. So in this case, we normally use a statistical software like Excel to fit the data to a linear regression. And the output is always in the equation given as y equals ms plus b. And this is normally called the equation of the line. So where the m is the slope or the gradient of the graph, the b is the y, the b is the intercept on the y-axis, the y is the measured signal or absorbance on the y-axis, and s is the known concentration on the s-axis. The sixth step is to check the linearity to obtain the coefficient of determination, which is the r square. So we obtain the coefficient of determination to assess the linearity of the calibration curve. And what we also have to note is that an r square value close to 1 indicates a good linear relationship. And we also check the linearity of our graph by using Excel. So the next thing to do is the interpolation of sample analysis. So first of all, what we have to note is that to determine the concentration of analyte, we measure the absorbance of the unknown sample at the required wavelength and use the calibration curve to interpolate the concentrations. And this can be done using the equation of the line as explained earlier, giving us y equals ms plus b, generated from the calibration curve, where m is the slope or gradient, b is the y-intercept, y is the measured signal or the absorbance, and S is the known concentrations on the S-axis. So let us take an example and see how we can interpolate or extrapolate the concentration from a calibration curve. So example, the measured absorbance of protein in an unknown sample at a wavelength of 760 nanometer is 45. Determine the protein concentration in the sample using a standard or a calibration curve. Yeah. So this is our calibration curve drawn over here nicely. So this is the instrument response, which is the same as the absorbance, and this is the concentration, and this is our line of equation, and this is our R square over here. So first of all, what we have to notice that from the calibration curve, the line of equation is giving us y equals 52.357 S plus 0.6286, where y is the absorbance on the y-axis, 52.357 the gradient or the slope of the graph, and S is the concentration on the S-axis, and 0.286, so the 0.6286 is the intercept on the y-axis. So what we have to do is to make S the subject from this equation. So making S the subject, we will have S equals y minus 0.6286, all divided by the slope, which is 52.357. So after this, we then put in the value of the absorbance, which is the y. So if you are to substitute 45 in place of y, so we will get 45 minus 0.6286, all divided by the slope. So that our final value for S will be 0.84 by. So what it means is that the concentration of the protein concentration of our sample is 0.84 per milligrams per liter over here. Let's look at some advantages of the calibration curve. So the first advantage has to do with accuracy. So calibration curves allow for accurate quantification of analyte in a sample by comparing the signal intensity of the unknown sample to the known standard. The next is sensitivity. So calibration curves can be used to determine the sensitivity of an analytical method, which is the ability to detect and measure small changes in the concentration of an analyte. And third is the linearity. So calibration curves provides a linear relationship between the signal intensity and the concentration of the analyte, which simplifies data analysis and interpolation. So this is a summary of the calibration curve over here. So this is our signal or the absorbances on the y-axis, and this is the concentration on the s-axis over here. So if we should look at the nature of the calibration curve over here, so we have our signal measured for the calibration curve, And this is our linear regression calculated or obtained from x, or in the form of the equation y equals ms plus c, where y is the absorbance, m is the slope, s is the concentration, and c is the intercept. And this is our coefficient, so which is the r square, which indicates how good or how best your graph is. And this is the intercept that gives an indication of the LOD in the graph. And this is our signal measured for sample of concentration. And this is the concentration of samples calculated from the equation of the line to be 3.5. Thank you.